or start or continue the next part of the human genome project. Earlier we discussed the definition of human genome project, right? Then why it is considered as mega project. Then we discussed the goals of human genome project is HGP and covered one of the method of the HGP. Now today we are going to discuss the second method of studying the human genome project, which is called as I am writing second method, sequence annotation method. In this method, the name of the scientist who worked, Sanger method or Sanger termination method. Sequences of DNA nucleotide. First the sequences occur, then work or special function are assigned after the sequencing. The Sanger in 1977 he worked on the sequence annotation method. First step is the extraction of the DNA from the source. And how it is extracted? This is the cell, it is the outer membrane, nucleus, DNA is there. First, by the enzyme, this plasma membrane is dissolved, then nuclear membrane, then DNA is taken up, isolated. Extraction of DNA. Then the second is requirement of the primers. Primers are the synthesized artificial DNA nucleotide which has in this particular use around 20 base pair, BP, 20 base pair. Then the third is the addition of nucleotides. Then the scientists pour all these chemical, DNA is also the chemical, in a test tube solution, and then the termination of DNA occurs. Termination means cutting of the DNA by the nucleotides, and it is cutting at the adenosine site, termination. Then termination means it breaks down into fragment. DNA breaks down into fragment, and then the scientists pour all this into the petri dish having the matrix of the gel. Now this DNA fragment after termination has different length, small, big, and appear to be dark. And this method is easy to sequencing the bases adenine, thymine, cytosine, guanine. This is the first important. Second important, it helps to study the familial variant. Variant means difference when there is any change or alteration in the DNA nucleotide bases running in the family, inherited. This condition is called familial variant, and this familial variant is due to the change, alteration of the DNA due to mutation or due to some pathogenic condition. So these are studied. That is the reason why Sanger method is considered to be more important method. Now, last lecture and today lecture complete the DNA or human genome project method. The next time we discuss the next part of the human genome project. Any question? Any query?
No, ma'am.
Thank you.